Okay, so welcome to this next video in which we are discussing the synthesis and secretion of insulin. Okay, right, so we've discussed that when blood glucose goes up, this will lead to a rise in the glucose concentration within the beta cell of the islet of Langerhans. Okay, and that an increase in the glucose concentration within the cytoplasm of this beta cell will lead to an increased rate of glycolysis, which will generate more ATP. So the level of ATP within the cytoplasm of the beta cell will go up. Okay, and it's this rise in ATP that is going to lead the beta cell to secrete vesicles containing insulin. Okay, and we're in the process of discussing how a rise in ATP is going to lead to a rise in intracellular calcium, and it's going to do it by working on ATP-sensitive potassium channels. Okay, so we've discussed that the ATP-sensitive potassium channels are a form of inward rectifying potassium channels, but they have a slightly more complicated structure than the normal inward rectifying potassium channels and the other ones that we've discussed. So let's discuss what's different about them. So let me draw a picture from above. So we have discussed that most inward rectifying potassium channels consist of 4 inward rectifying potassium channel subunits stuck together in a tetramer like so. And basically ATP-sensitive potassium channels do have this, but then they have other proteins attached around the inward rectifying potassium channel subunits. Okay, so let's now discuss these other proteins that will attach to the inward rectifying potassium channel subunits, and one of these proteins is going to attach to each one of the inward rectifying potassium channel subunits. Okay, so these are now inward rectifying potassium channel subunits that are in family 6, so I'll put 6.X, so it's either 6.1 or 6.2, and then surrounding it that is bound to this inward rectifying potassium channel subunit, this is what is known as a sulfonylurea receptor. Okay, and for short, sulfonylurea receptors are abbreviated to SURs. Okay, so this is usually abbreviated to an SUR. Now, let me show you the structure of a sulfonylurea receptor subunit. So we have seen, we've seen the structure of a inward rectifying potassium channel subunit. So each of the inward rectifying potassium channel subunits have a membrane spanning topology like so. Okay, so their amino and carboxy termini are both on the cytoplasmic side, and they have 2 membrane-spanning alpha helices, and they have this P-loop in between the 2 membrane-spanning alpha helices. Now let's have a look at the membrane-spanning topology of a sulfonylurea receptor. So, sulfonylurea receptors are what are known as an atypical ABC protein. Okay, so most ABC proteins are transporters. Sulfonylurea receptors are not going to be transporters. And by the way, ABC stands for ATP binding cassette. Okay, so basically the sulfonylurea receptors are members of this family of proteins known as the ATP binding cassette proteins. Okay, or the ABC proteins. They are atypical ABC proteins because they're not going to be involved in the transport of molecules across the plasma membrane. Okay, so let me now show you the membrane spanning topology of a sulfonylurea receptor, and it's a big protein. Okay, so here is the plasma membrane. The amino terminus starts off in the extracellular fluid over here, like so. Then you have 5 membrane-spanning alpha helices, 1, 2, 3, 4, 5, okay? Then you have another 6 membrane-spanning alpha helices, all together like so. Then you have a very special domain called the nucleotide binding domain 1. Sorry about that. Okay, and then you have another region of 6 membrane-spanning alpha helices here, like so. And then finally another nucleotide binding domain here known as the nucleotide binding domain 2, and then you have the carboxylic acid group here. Okay, right, so let's label the bits of this protein up then. So this portion over here, this portion of 5 membrane-spanning alpha helices, which are all close by one another, this is known as the transmembrane domain naught. Okay, so the TMD naught. Now this is a special portion that normal ABC proteins don't have, but sulfonylureas all have. Okay, so the normal structure of a typical ATP binding cassette protein is that you have these two here, these two groups of 6 membrane spanning alpha helices with the 2 nucleotide binding domains like this, but this extra 5 membrane spanning alpha helices, which are all grouped together, this is abnormal. So this is something that's special for the sulfonylureas. Okay, so TMD0 stands for transmembrane, so the T is for trans, the M is for membrane, and then the D is for domain, and that's the transmembrane domain 0. Okay, now this next transmembrane domain here, consisting of these 6 membrane spanning alpha helices, this is known as the transmembrane domain 1. So this is TMD1. Okay, and then the next one here, which I have in pink, this group of 6 membrane spanning alpha helices, this is known as transmembrane domain 2, or TMD2. Okay, and the other important things that we have are these 2 special domains here, and these are the nucleotide binding domains, or NBD for short. So this is the NBD1, so this stands for nucleotide binding domain 1. Okay, and these nucleotide binding domains, they will bind ADP in the case of the sulfonylurea receptors. So this is the nucleotide binding domain 1, and here this is the nucleotide binding domain 2. Right, so this is the structure of a sulfonylurea receptor. And as I say, the transmembrane domain naught is the abnormal bit. This bit, the transmembrane domain 1, the nucleotide binding domain, the transmembrane domain 2, and then another nucleotide binding domain, this is very, very typical ATP binding cassette protein structure. This is the odd bit over here. And it's this bit over here, this transmembrane domain naught, which is involved in binding to the inwardly rectifying potassium channel subunit of the 6th family. Right. Okay, so how many sulfonylurea receptors are there? Well, basically there are 2 genes which code for the sulfonylurea receptor proteins. Okay, so these are the sulfonylurea receptor 1 protein, and then there's also the sulfonylurea receptor 2 protein. So these 2 separate genes which code for different sulfonylurea receptor proteins, okay? And the gene that codes for the sulfonylurea receptor 1 is known as ABCC. So CC8. And the gene which codes for the sulfonylurea receptor type 2 is known as the ABCC9. Now, when you produce the sulfonylurea receptor 2, there are 2 possible splice variants. So from this same gene, you can get 2 different proteins by having 2 different splice variants. And these are known as the sulfonylurea receptor 2A and also the sulfonylurea receptor 2B. Okay, right, so there are overall 3 different sulfonylurea receptors that you can produce. There's the sulfonylurea receptor 1, which comes from the ABCC8 gene. Then there are the 2 splice variants of the sulfonylurea receptor 2, the sulfonylurea receptor 2A and the sulfonylurea receptor 2B, which both come from this gene ABCC9. Okay, right, so let's now talk about the form that is in pancreatic beta cells, okay? But just before we talk about that, actually, I want to continue talking about ATP-sensitive potassium channels in more general terms, okay? So basically, ATP is going to bind directly to the inward rectifying potassium channel subunits. So ATP within the cytoplasm of the cell will bind to the cytoplasmic domains of these inwardly rectifying potassium channel subunits and causes the channel to close, basically. So it promotes the channel being closed. Meanwhile, if ADP goes up in the cytoplasm, ADP will bind to the sulfonylurea receptors through the nucleotide binding domains, and then the sulfonylurea receptors will interact with the inwardly rectifying potassium channel subunits of the 6th family and cause the channel to open. So what does this mean? It means that when there is very high ATP in the cell, obviously that means there'll be less ADP, because in order to make ATP, you take the ADP and add a phosphate group onto it to create ATP. So when ATP is high, ADP will be low. ATP will be it will be acting on the inwardly rectifying potassium channel subunits and causing them to close. There'll be less ADP telling the sulfonylurea receptors to tell the inwardly rectifying potassium channel subunits to open, so that's going to mean that the channel closes when you have very high levels of ATP. So when you're doing metabolically well, the channel will close, and when you're doing metabolically badly, what will happen is you'll have high levels of ADP, low levels of ATP, which means that the ATP won't be telling the inwardly rectifying potassium channels to close, and instead the ADP will be binding to the sulfonylurea receptors, which will then tell the inwardly rectifying potassium channels to open. So the channel opens when you've got high levels of ADP and low levels of ATP, i.e. when you're doing metabolically badly. Okay, right, so let's now talk about the form of this that you have within the pancreatic beta cells. So, the specific form that you have within the pancreatic beta cells is made up of a tetramer of KIR 6.2 subunits. So remember, in the 6th family of inwardly rectifying potassium channels, there were 2 genes, basically. There was the KIR 6.1 and the KIR 6.2. So you're going to use 4 KIR 6.2 proteins to make a homotetramer, okay? And this will be your inwardly rectifying potassium channel. And then surrounding these inwardly rectifying potassium channel subunits, you're going to have 4 sulfonylurea receptor 1s, okay, and this will make an octamer. Okay, so this is the form of the ATP-sensitive potassium channel that you have in pancreatic beta cells. You have the inwardly rectifying potassium channel subunit 6.2 with the sulfonylurea receptor 1. Now, how is this going to lead then to the release of insulin? So, basically, if we draw the membrane, so here is our membrane, and then we have this massive octamer here, and I'll draw this out. Okay, so here we have the octamer, here's the channel right in the center, and whoops, let me draw this. So these are the inner layer, which is made up of the inwardly rectifying potassium channel 6.2 subunits. Okay, and then the outside is made up of these sulfonylurea receptor 1 proteins, like so. Okay, so here is our ATP-sensitive potassium channel that is in the membrane of the beta cells of the islets of Langerhans. So, what's going to happen when ATP goes up because glucose concentration in the cytoplasm of the beta cell has gone up? Well, basically, ATP is going to bind to the inwardly rectifying potassium channel 6.2 subunits, and it's going to cause the channel to close. Now, usually, it's open, okay, so it'll spend some time open and sometimes closed, but once the ATP goes up, a majority of the K ATP channels will now close, basically. So this is a K ATP channel. So more of them are going to be closed at any one time, which means that the amount of potassium they are going to be allowing to move out of the cell is going to go from being, let's say, this size, and it's going to move down to a much smaller size. So remember, we'll have loads of these channels in the cell membrane, okay? So if most of them are now, if a good few of them are now closed most of the time, then that means that the overall current of potassium ions that we're going to allow to move out of the cell through these channels is going to decrease. Now, let's just have a reminder of what resting membrane potential across the cell membrane is and how it works. So, if I draw my beta cell here, then basically what we need to know is the potassium and the sodium concentration gradients across this cell membrane. So we'll start off with the sodium concentration gradient. Sodium concentration extracellularly is around 145 millimolar, whereas intracellularly it's around 12 millimolar. Okay, we know that potassium concentration then intracellularly is around 155 millimolar, and extracellularly it's around 4 millimolar. Now, basically, the resting membrane potential across the cell membrane of a beta cell of the islets of Langerhans is usually around negative 60 millivolts, which means that usually the electrical potential of the intracellular compartment is 60 millivolts lower than the electrical potential of the extracellular compartment. Now, this is the resting membrane potential, or the equilibrium potential. Now, why is it the equilibrium potential? What does that actually mean? It means that it is the electrical potential that you need to have across that cell membrane so that there is no net movement of charge across that membrane. It is not, repeat, it is not the Nernst potential for sodium or the Nernst potential for potassium. So at this potential you will still have net movements of sodium and net movements of potassium across this membrane. It's just that the movement of sodium ions into the cell will balance the movement of potassium ions out of the cell so that you get no net movement of charge across the membrane, and therefore the electrical potential difference across the membrane doesn't change. So we're talking now about resting potential, not Nernst potentials. Okay, right. So, let's think this through then. So, at this resting potential, what we know is true is that the movement of charge is balanced, basically. Now let's think through what's going to happen to the sodium ions. So basically, the sodium concentration gradient is favouring the movement of sodium into the cell. In addition, sodium is a positively charged ion, so it wants to go to the place where the electrical potential is lower, which is the intracellular compartment, which is currently lower by 60 millivolts than the extracellular compartment. So this means that both the concentration gradient and the electrical gradient are favouring the movement of sodium in. However, you do not have a large permeability to sodium across the membrane. The permeability to potassium ions is much larger, so we've got a little bit of a current of sodium into the cell. Okay, and note that this is at resting potential. You have got a net movement of sodium into the cell, so the sodium concentrations will not be at equilibrium in the at the electrical equilibrium point. Okay, now let's think through potassium. So, we've got a very high concentration of potassium in the cell, a lower concentration extracellularly. The concentration gradient is therefore telling potassium to move out, but the electrical gradient this time is in opposition. The electrical gradient is favouring the movement of potassium in, because potassium again is a positively charged ion and will want to go where the electrical potential is lower, which is the intracellular compartment. Okay, but we know that the concentration gradient is easily going to win. Remember, the electrical gradient needs to be negative 85 millivolts before it will be strong enough to oppose completely this concentration gradient. So you're still going to get a movement of potassium out. And the permeability to potassium ions is much higher in the case of potassium than it is for sodium. So actually the movement of potassium ions is exactly opposite to the movement of sodium ions. Okay, so this means that the electrical the movement of charge is completely balanced, because if we've got a certain movement of sodium ions in and the exact same movement of potassium out, they both carry one positive charge, so that balances the movement of charge. You've got no net movement of charge across the membrane, and that's why this is at equilibrium, or at least electrical equilibrium, because there's going to be no movement of charge and therefore no change to the electrical potential difference across the membrane. Okay, right. Now you might say, well, there's a problem here. the cell is going to fill up with sodium and it's going to lose all its potassium, but then that's the purpose of the sodium-potassium pump. Okay, so the sodium-potassium pump then reverses the movement of sodium and potassium. So it then chucks sodium out of the cell and moves potassium back into the cell. Okay, so it chucks sodium out and potassium back in, and it uses ATP to do that, and that's how you keep the concentrations of sodium and potassium the same, because at equilibrium potential, the concentrations are going to change if you don't have that, because you've got a net movement of sodium in and a net movement of potassium out. And if you don't have something that's reversing that net movement of the ions, then the ionic gradients are going to be destroyed. Okay, so that's the purpose of the sodium-potassium ATPase. Okay, so this is the sodium-potassium pump. Right, okay, so now we can talk about these K-ATP channels and what the effect of closing them is going to have. So basically, these K-ATP channels contribute to the permeability of potassium. They're not the only type of potassium channel that's usually open in the cell membrane, but they are one of them. So the way that the potassium ions are moving out is through these channels, and also other types of channels, but one of the means by which potassium channels are one of the means by which potassium ions are moving out of the cell is through these ATP-sensitive potassium channels. Now, when we close a good majority of these, what will happen is that permeability to potassium will go down, and that means that this net movement of potassium across the membrane is going to go down because the permeability has gone down, and suddenly we are going to break the electrical equilibrium, because now the sodium movement into the cell is greater than the potassium movement out of the cell. So you're going to get a net movement of positive charge into the cell. And if you move positive charge into the cell from the extracellular fluid, that means that you are going to raise the electrical potential of the intracellular compartment, because you're sticking positive charge there. So the electrical potential intracellularly will be going up. Okay, so this goes up. And it also means that you're going to be reducing the electrical potential extracellularly, because you're moving positive charge out of the extracellular fluid. Now, if we remember what the voltage across the membrane was, it was the electrical potential difference from extracellular to intracellular. It was how much the electrical potential changes if you move from extracellular to intracellular. Originally, it went down by 60 millivolts, okay? So this one was 60 millivolts lower than this one, but if we're moving this one down and we're moving this one up, then the amount by which the intracellular electrical potential will be lower than the extracellular electrical potential will go down. So this number is going to become less negative, basically, i.e. the membrane is going to be depolarized. Okay, so mathematically we can see this because the electrical potential difference was the electrical potential intracellularly minus the electrical potential extracellularly. This number's going down, this number's going up, so this number is going to become less negative, basically. Okay, and that's known as a depolarization. And when we depolarize the electrical potential difference across this cell membrane of this beta cell of the islets of Langerhans, this is going to activate voltage-gated calcium channels of the L type, and that's going to allow calcium to move into the cell. Okay, so we'll now just have a quick discussion of L-type voltage-gated calcium channels, but in fact I think we'll do that in the next video.